Welcome back to Chapter 16, the citric acid cycle. In part 2 of this chapter, we will discuss reactions of the citric acid cycle. Reactions of the citric acid cycle are summarized in this slide. As you can see, there are 8 different reactions that happen in this cycle. Citric acid cycle is where acetyl-CoA gets oxidized to 2 carbon dioxide molecules, and it yields 3 NADH molecules along with FADH2 and a GTP molecule. Although citric acid cycle is central to yielding energy, its role is not limited to energy conservation. There are various different metabolites, 4 and 5 carbon intermediates in this case, that emerge from this cycle at various different points and various different steps. These intermediates serve as precursors for a wide variety of metabolic products, and hence TCA is central to various different metabolism. The first step of citric acid cycle involves the conversion of acetyl-CoA to citrate. In converting acetyl-CoA to citrate, the enzyme citrate synthase uses another intermediate, oxaloacetate. This involves condensation of acetyl-CoA and oxaloacetate, and this is the only reaction in citric acid cycle that forms a C-C bond. This enzyme uses acid-base catalysis. As you know, carbonyl of oxaloacetate is a good electrophile, and methyl of acetyl-CoA is a good nucleophile because it's next to a carbonyl carbon, and this methyl group, the hydrogen in this methyl group, is acidic. So there is an aldol condensation-like reaction that happens to form citrate. Rate-limiting steps of TCA cycle is conversion of acetyl-CoA to citrate. The activity of the enzyme citrate synthase largely depends on the concentration of oxaloacetate. Finally, the reaction is highly thermodynamically favorable and practically irreversible, as you can see from the delta G0. It's regulated by substrate availability and property. Let us consider the citrate synthase reaction mechanism. Once both substrates, oxaloacetate and acetyl-CoA, binds to the enzyme active site, a base, aspartate 375, in the active site of the enzyme abstracts a proton and forms an enolate intermediate like this. And the carbonyl then abstracts a proton from histidine 274 to which it hydrogen bonds. Once the enol intermediate is polarized, electrons are pushed back, and this acts as a nucleophile to attack this carbonyl of oxaloacetate, which then takes up a proton from adjacent histidine, which acts as a general acid. Once this step happens, the oxaloacetate and the enol intermediate are converted to citral coenzyme A. And as you can see, this carbonyl is still hydrogen bonded to histidine 274. In the further reaction, water in the active site hydrolyzes the thioester, which is the citral coenzyme A, and pushes out the coenzyme A. This results in the formation of citrate, and hence the reaction between oxaloacetate and acetyl-CoA results in citrate. Induced fit drives the reaction catalyzed by citrate synthase. This enzyme, as you can see, exist in two different conformations. One is an open conformation for free enzyme that does not have a binding site for acetyl-CoA, whereas a closed conformation binds both acetyl-CoA and oxaloacetate. The way this enzyme works is like this. In the presence of both oxaloacetate and acetyl-CoA, the enzyme first binds oxaloacetate, as a result of which there is induced fit and conformational change, which further allows the enzyme to bind the acetyl-CoA. One of the reasons why induced fit happens is to avoid unnecessary hydrolysis of acetyl-CoA. Because if acetyl-CoA first binds in the active site, water in the active site can hydrolyze the CoA, the thioester in acetyl-CoA, resulting in just acetate. That can hamper the whole sequence of reactions. Step 2 involves conversion of citrate to isocitrate. Formation of isocitrate from citrate is catalyzed by the enzyme aconitase. Elimination of water, as shown here, from citrate gives a CC double bond containing compound called cis-aconitate. Water is then added across this double bond stereospecifically to form isocitrate. So, in a nutshell, the enzyme aconitase catalyzes a dehydration as well as a rehydration reaction to convert citrate to isocitrate. Addition of water to cis-aconitate is stereospecific. And most importantly, the standard free energy change is positive, and hence the reaction is unfavorable and reversible. But the product concentration is kept low to pull the reaction forward, which means that isocitrate is constantly carried over for the next reaction. The dehydration and rehydration of citrate to isocitrate is catalyzed by the Iron-Sulfur Center in the enzyme aconitase. The Iron-Sulfur Center is shown in red here. 3 cysteine residues, 1, 2, and 3, in the active site of the enzyme bind 3 iron atoms, 1, 2, and 3. The 4th iron is bound to one of the carboxyl groups, as shown here, of citrate, and also interacts non-covalently with the hydroxy group of citrate, and shown by this dashed line. A basic residue, in this case, in the enzyme, helps to position the citrate in the active site. The Iron-Sulfur Center acts in both substrate binding and catalysis. In step 3, isocitrate is converted to alpha-ketoglutarate. In step 3, oxidative decarboxylation of isocitrate happens, and this is catalyzed by Isocitrate Dehydrogenase. During this reaction, isocitrate is first oxidized to oxalosuccinate by the enzyme using NADP as a cofactor. Oxalosuccinate then undergoes decarboxylation and rearrangement of the enol to form alpha-ketoglutarate. The first step is the oxidation of alcohol to a ketone, and it involves a transfer of a hydride from isocitrate to NADP. There are 2 different forms of isocitrate dehydrogenase enzyme. One is a cytosolic enzyme that uses NADP+ as a cofactor. This enzyme can also use NAD+ as a cofactor. The other isoform is a mitochondrial enzyme, which uses NAD+ as a cofactor. This is a highly thermodynamically favorable reaction, going from isocitrate to alpha-ketoglutarate. and it is regulated by product inhibition and ATP. In step 4, alpha-ketoglutarate is converted to Succinyl-CoA. Oxidation of alpha-ketoglutarate to Succinyl-CoA is catalyzed by the enzyme alpha-ketoglutarate dehydrogenase. This happens to be the last oxidative decarboxylation step. Also, net full oxidation of all carbons of glucose has been accomplished. And this has been accomplished after 2 turns of the cycle. However, carbons are not directly from glucose. This is because this carboxylate came from oxaloacetate. If you remember, the first intermediate, citrate, was formed by combining Acetyl-CoA and oxaloacetate. And so this carboxyl is originally from oxaloacetate, and that's the carboxylate that gets removed as a carbon dioxide. The reaction catalyzed by this enzyme complex, that is alpha-ketoglutarate dehydrogenase complex, is very similar to the pyruvate dehydrogenase complex that we talked about in the previous lecture. The mechanism of the reaction is also very similar. The product of the reaction, Succinyl-CoA, is an intermediate that has a high energy thioester bond, as you can see here. And this reaction is highly thermodynamically favorable and nearly irreversible. This enzyme is regulated by product inhibition as well. In step 5, Succinyl-CoA is converted to succinate. Conversion of Succinyl-CoA to succinate is catalyzed by the enzyme Succinyl-CoA Synthetase. And this reaction involves substrate level phosphorylation, which means that in this reaction, a GDP molecule is converted to a GTP molecule with the release of coenzyme A. The energy of thioester bond in Succinyl-CoA allows for incorporation of inorganic phosphate onto GDP to make GTP. Also, this reaction goes through a phosphoenzyme intermediate. Furthermore, this reaction produces GTP, or guanosine triphosphate, which can be converted to adenosine triphosphate, or ATP. This reaction is slightly thermodynamically favorable, and it can be reversible if required, with a standard free energy change of minus 2.9 kilojoules per moles. One way in which cells keep the reaction towards the right is by keeping the product concentration low, which means that succinate is constantly channeled to the next reaction. In step 6, succinate is converted to fumarate. The conversion of succinate to fumarate is catalyzed by the enzyme Succinate Dehydrogenase. This enzyme is bound to the mitochondrial inner membrane, and hence this reaction does not happen on the mitochondrial matrix. This enzyme is a part of complex 2 in the electron transport chain. It'll be clear to you when we discuss oxidative phosphorylation as to what complex 2 does in the electron transport chain. Furthermore, FAD is used as a cofactor by this enzyme to oxidize succinate to the alkene derivative fumarate. FAD is usually covalently bound to the enzyme, and more importantly, the delta G0 is 0 kilojoules per mole, which means it's kept at equilibrium and can be reversible. product concentration is usually kept low to pull the reaction forward. So what is FAD? We have seen this molecule briefly in chapter 13. If you recollect, FAD stands for flavine adenine dinucleotide. And as you can see, this is the business end of the molecule, which is called as the isalloxazine ring. And when FAD gets oxidized, gets reduced to FADH2, the structure changes from here to here. Now, one of the characteristic features of FAD is that it can do 1 electron reduction or 1 electron oxidation. NAD, on the other hand, can only do 2 electron oxidation or 2 electron reduction. In step 7, fumarate is converted to L-malate. Conversion of fumarate to L-malate is catalyzed by the enzyme Fumarase. And this enzyme is stereospecific, which means that the addition of water to fumarate is always trans, and it results in the formation of L-malate. In the first step, OH- is added to fumarate, and that is across the trans side, and then a proton is added to the carbanion that is formed as a result of the addition of hydroxy to the double bond. This reaction is slightly thermodynamically favorable, and it is close to equilibrium, as you can see, and hence can be reversible. One way in which the cell pulls this reaction forward is by keeping the concentration of L-malate low. Step 8 involves the conversion of L-malate to oxaloacetate. Conversion of L-malate to oxaloacetate is catalyzed by the enzyme Malate Dehydrogenase. And this is the final step of the tricarboxylic acid cycle. This step regenerates oxaloacetate for the enzyme citrate synthase. If you remember, citrate synthase is the first enzyme of the TCA cycle. This reaction is highly thermodynamically unfavorable, as seen from the standard free energy change of 29.7 kilojoules per moles, and is reversible. The oxaloacetate concentration is kept very low by the enzyme citrate synthase, which pulls the reaction forward because oxaloacetate is constantly used up by this enzyme citrate synthase to convert acetyl-CoA to citrate. And hence the final reaction is pulled forward and the cycle continues. So to recap, citric acid cycle or tricarboxylic acid cycle, or the Krebs cycle, you may call it any of these names. This cycle involves the conversion of acetyl-CoA into 2 carbon dioxide molecule and eventually to oxaloacetate. And in this process, 1, 2, 3 NADH molecules are obtained from 1 molecule of acetyl-CoA along with 1 FADH2 and 1 GTP. So the yield that one gets from 1 acetyl-CoA molecule is 3 NADH, 1 FADH2 and 1 GTP. Before moving ahead, let us take a step back and look at the direct and indirect ATP yield from both these processes. The first process is glycolysis, which converts glucose to pyruvate. And the second process is when pyruvate gets converted to oxaloacetate while going through the citric acid cycle. Now, going from glucose to glucose 6-phosphate or fructose 6-phosphate to fructose 1,6-bisphosphate in the preparatory phase is when 2-ATP is invested by the cell. And during the payoff phase, glyceraldehyde 3-phosphate to 1,3-bis-phosphoglycerate, and in this case 2 molecules of glyceraldehyde 3-phosphate catalyzed by GAPDH, and in further steps 4 ATP molecules are produced along with 2 NADH. So the net yield there, as we talked about, is 4 minus 2, so 2 ATP plus 2 NADH. Now, pyruvate doesn't stop there, right? There are 2 pyruvate molecules that enter the citric acid cycle. Now, what we talked about in the previous slides is the conversion of 1 pyruvate molecule to oxaloacetate. And in this process, 3 NADH molecules, 1 FADH2, and 1 GTP is produced. Now, if you consider 2 pyruvate molecules, and we have not considered the first step in the conversion of pyruvate to acetyl-CoA molecule. So if you consider 2 pyruvate molecules that gets converted to 2 acetyl-CoA molecules where 2 NADH is produced, that also adds to the equation. Now, starting from the isocitrate to alpha-ketoglutarate reaction, TCA cycle starts, right? So 1, 2, and 3 NADH molecules from each pyruvate. And if there are 2 pyruvate, that's 2 plus 2 plus 2. So 6 NADH molecules, 2 GTP molecules, and 2 FADH2 molecules, along with the 2 NADH that is obtained in the beginning, where pyruvate dehydrogenase complex converts pyruvate to 2 acetyl-CoA molecules. Now, if you convert NADH, GTP, and FADH2 in terms of ATP equivalents, and 1 FADH2, I have not told you this before, 1 FADH2 equals 1.5 ATP molecules. So 2 FADH2 in principle corresponds to 3 ATP molecules. Now if you combine all these ATP molecules formed, starting from the glycolysis reaction, and consider the investment, the total number of ATP produced is about 30 to 32.